Okay, so next talk is about biophotonics, as the title would suggest. Now the reason I want to do this in this order is hopefully kind of clearer now, in that optofluidic technology really provides a nice home for doing this kind of biophotonic sensing, because most of the bio stuff comes in water. What's water? Liquid. If we want to sense stuff in liquid, we use optofluidics. So, to introduce my talk in the same way that I introduced my last talk, what do I mean by biophotonics? Biology plus photonics. Now, my last job was in Boston in a Department of Biomedical Engineering. And working there really sort of opened my eyes to the possibilities available for using photonic technologies in the bioengineering, biomedical realms. Now, painful though it may be to speak to bioscience people who really do speak different languages, us physical sciences type and bioscience people, once you get on the same page, and once you get them understanding that they can do a lot of their light-based stuff a lot better using technology that's been out there in photonics for decades, progress can really be made. And it's, in my humble opinion, that this will be one of those next hot fields where there are easy pickings. These strange noises in this lecture are quite confronting. So, yeah, as I said, biophotonics, from my perspective anyway, is getting all this stuff happening in biology, working on this vast base and body of work that biosciences have been developing for yonks, and applying our photonic technologies to it. Now, biophotonics is actually a little bit more common than I would have you believe. Now, I used a very dodgy example of a contact lens. It's not really photonics, it's kind of bulk optics, but that's all right. More seriously though, the blood glucose monitor is an example of a very successful, very accurate biophotonic device. Sure, you're only sensing one thing, sure it takes a bit of time, but a lot of the devices still use this colorimetric response whereby an enzyme on your little strip reacts with the glucose in your blood, changes colour, and tells you how much there is. Relatively, well, I was going to say barbaric, but relatively primitive anyway. Speaking of barbaric, photonics has actually had a long history with medicine. This is a medieval document charting urine colour and supposedly telling you precisely what's going on inside the patient. I don't even know what language that's in, but I'm sure it was wildly inaccurate as to the biological process going on. The point being that health, medicine, and some kind of optics in diagnosis have had a long partnership and a long history. And hopefully we can do better than what we already have. So, okay. I'm going to cover a tiny, tiny subset of topics available in biophotonics. I'm going to very well define what this talk isn't about, and then I will define what this talk is about. So, when most people mention biophotonics, the mind almost immediately goes to light microscopy. And don't get me wrong, advances in light microscopy have been amazing over the last decade. The specific binding of various fluorescent dyes, stains, quantum dots, that sort of thing. We have unparalleled imaging, detection, resolution of what's going on inside cells. This has gone even further to be able to specifically bind fluorescent molecules to individual chromosomes in DNA. This I also find absolutely amazing. The fact that you can just look under a light microscope, look at this fluorescent particle and tell what is going on in somebody's DNA. Very powerful technique, and it's coming to the fore in commercial pathology, for instance. Tell what precise genetic conditions you have. And of course we have the Nobel Prize winning green fluorescent protein, whereby mitochondria in cells can be genetically engineered to express different colours of fluorescent molecules, and you can image these highly specific fluorophores in living cells. And rightly so. This work, as I think I mentioned, won the Nobel Prize, if not the most recent one and the one before. I am not going to talk to you about microscopy. As I said, microscopy, responsible for many of the great advances in the bioscience. What I am going to talk to you about is photonic design applied to biological materials for sensing and biomedical engineering. On the right there is another thing you can't see, don't worry about it. Now, I mentioned several times in my last talk about functionalizing, so adding very specific binding molecules to photonic structures. So now here's the ring, here's some stuff, and here's some stuff binding to it. And we see a shifting wavelength that tells us there's stuff. That's pretty cool, but it's also pretty hard. The kind of chemistry involved is what's called cyanization. So you have to take these silicon molecules, you have to make one end acceptable to bind onto the material of your photonic device, so a glass, semiconductor, something that's very non-bio, shall we say, and you have to make the other end very friendly to bio. It's difficult chemistry. And I'm not a chemist, I can't do any of this. So I'd like to do something that gets around that. That becomes something the materials problem. And I found that the material was silk. Same stuff that your shirts, your ties, and possibly your underwear, if you're into that sort of thing, are made out of. So, let me tell you a little bit about the material, what we can do with it, and hopefully I'll convince you that silk is actually like a really good biophotonic material. So, we start off with the cocoons of the Bombyx morai silk caterpillar. That's those things up there. We kill the little worm inside, and then we boil down the fibre in salt, the fibres in salt. We dialyze this solution, so that gives us a selectivity over what weight of molecule we can extract. We do this properly, and that gives us an aqueous solution of silk protein. We can then cast that silk protein onto a structure, let it dry out, and we're left with a film of silk which can contain spatial modulations, such as this hologram of my previous employer's logo. Now, what we get out of the silk fibres, there are two distinct proteins in the silk fibres. There's stuff called fibrin and stuff called sericin. Now, you may have all heard of silk's legendary strength as a natural material and luster. The strength is provided by the fibrin molecules. The sericin provides a kind of glue, so that provides the elasticity of the thread, of the naturally occurring thread. And all silks contain these two molecules to different amounts. Spider silk contains more sericin, so that it's more elastic and can actually trap things. The Bombyx morai silk is just meant to be strong and protect the larva. Doesn't work from a nail to the head, but that's all right. So we have these fibrin films. What are they like? Well, with our refining process, as it turns out, they're ridiculously clear. Far clearer than one would expect from any biomolecule in fact. You get almost 95% transmission in the visible, as you can see, clear as. In the infrared, surprisingly, aside from these water absorption features, which you can get rid of by appropriately dehydrating the film. And I'll get back to the dehydration I'll get to the dehydration in the next slide. You have something like 80% of transmission out into the mid-infrared. This is astounding for a biomolecule. Don't really know why it's so clear. It just is for now. Also has this very interesting conformal property, whereby if you just dry the silk out slowly from the aqueous solution, it naturally forms into these alpha helical structures, we believe. If you dry out the silk film, if you dehydrate it using alcohol, soap, you can transform it into this beta sheet structure. Now what's interesting about this is that these two conformations have astoundingly different solubilities. That means you can, by controlling the beta sheet content in your silk, you can actually control the solubility of your device. Say you wanted a medical diagnostic chip that would sit in a human being for a matter of days while they were being treated in hospital and then just dissolved into the bloodstream. The other thing you can do with silk that I'm going to hint at now and then we'll revel in its full glory later is you can actually embed biomolecules into the silk. Now you don't actually have to do any complex biochemistry. Remember, I'm not a chemist, can't do any of this. We're taking horseradish peroxidase, which is an enzyme that has a colour change, related to ambient pH. Simply mixed into the silk film, dried it out, and then dried it out further to make it insoluble. And then dipped it in various pH solutions. And lo and behold, the enzyme changed colour. Now, this isn't particularly amazing with horseradish peroxidase. It's quite a robust enzyme. You can just keep it out on the shelf. But the fact you can mix it in to a substrate and have it stay active and reversible kind of gave us the idea at the time that we could do other things with it. And I'll get to that later. Perhaps one of the other bizarre characteristics of this material is the quality with which you can form it. We cast a silk film on a silicon wafer, cleaved silicon, so atomically flat, and this relatively large biomolecule actually reproduced the flatness of the surface to something under 5 nanometers root mean square of roughness. That's pretty amazing for a biomolecule, indeed for any kind of polymer or plastic. Again, why it does this, we don't know. It can also conform. This is an optical grating, which has a period of, what is it, some microns. And again, we gave excellent fidelity in reproduction of this structure by simply casting onto the grating. So we can play all sorts of games with this now that we know we can pattern it. We've made various lenses, diffraction gratings, holograms, lens arrays. Ultimately useless things, but fun nonetheless, and make very good photographs. The patterning process is surprisingly easy. Apart from casting, which runs the risk of having the material adhere to your template and removing it, which is bad. What we found was there's a certain point in the phase diagram of these fibers where we can adjust the humidity by a little bit, adjust the heat by a little bit. So you're talking, say, 40 degrees and just a little drop of water on your film, and then applying pressure. We call this technique contact lithography, and it's been very successful. To the point where we can actually pattern these aperiodic crystal structures with fidelity that you can't see again. Awesome. But anyway, what this shows is that you can get sub 5 nanometer resolution of simply imprinting these arrays of chrome dots onto your silk film. So that's sort of what the material can do, and you're all highly excited about it, I can see. What I'm going to show you next is some devices, preliminary though they may be, photonic devices that we have demonstrated in this material. Will hopefully show you the versatility as a photonic material. So the first thing we did was build a robot, like any good scientist does. And we built this robot to print waveguides. Relatively simple method. What we do is we have a glass slide, we have a drop of methanol, so remember I said that alcohols dehydrate the silk and make it tough, kind of set on the surface. and we have a silk solution that's being pumped through a pipette tip. It sits just above the surface of the slide and makes little waves or straight lines, and we couple it to it using the standard optical coupling techniques. And verily, it guides light. Beautiful. You can make these structures surprisingly smooth. Again, we weren't quite sure if you'd be able to print structures as smooth as we could make like casting, but it turns out that you can. There's something like 20 nanometers root mean squared of roughness on those waveguides. Still relatively high loss, but there's still a lot of work to be done in purification of the fibre. The salt separation process that I described earlier is actually quite rough on proteins. You can split them up into sort of a broad range of complex monomers whose behaviour when you dehydrate them is not entirely known. So there's a little cross section, and it's full of light. Yeah. Turns out we can also make it gain. Now, rhodamine is probably the most robust and ubiquitous laser dye, but we mixed it in anyway, and it fluoresced, which is, again, not entirely expected. The hypothesized chemistry of fibrin is quite complex. It's made up of something like 5,000 peptide chains, peptide molecules, with very variable hydrophobicity and hydrophilicity along the length of the chain. And it's this variation in surface chemistry that leads to the rich variety of behaviours that we think we see out of these materials. How that would interact with a fluorescent dye, who knows? Turns out they do fluoresce with good efficiency, and some other work has shown that they fluoresce orthogonally, which means you can mix a couple of fluorescent species in there, and they statistically pick up whatever light they're pumping. Again, not entirely known in biomolecular systems. So what have we done with it? We've made a pH sensor. Again, not entirely revolutionary, but as it turns out, we can functionalise a part of the silk chain so that we actually get what's called azide flipping. So in other words, we get an azide molecular attachment to part of the silk, and as pH changes its conformation, changes its optical properties. So as we apply basic pH, it goes red. We made a little microfluidic flow cell detector type thing, shown here, where we have a region of silk underneath this microfluidic channel, and as we flow things over it, it changes colour, which is what one wants out of a sensor. More specifically, it behaves like this. Perhaps the most interesting feature of this response is that you can apply very basic solutions to this functionalized silk without it degrading, and you can have the silk perform reversibly in terms of the pH in terms of the optical response to the pH you apply. This is also somewhat unexpected. Most biomolecules don't like base; they turn into salt. This one, robust, doesn't like acid though, which is interesting. Don't know why that is. That's why it's recent. So this leads me to my description of my latest device that I made out of silk. And this one, I hope, will give you something to particularly think about. I took my own hemoglobin, lysed it from my blood, and mixed it into a silk solution. No chemistry, just mixed it. I cast it onto a gradient in the hope that my hemoglobin would remain active, binding to ambient oxygen or releasing oxygen in the lactate role, change the spectral response of the very hemoglobin-infused gradient that we're using to analyse the light spectrally. The fabrication process was relatively straightforward. Like I said, silk, hemoglobin, add it to a grating, wait for it to dry, lift it off, and we're left with that rainbow-coloured piece of blood. Now, as I mentioned before, this silk can pattern optical structures with high fidelity, so it actually got very good optical efficiency out of the scrotum. We dried it out in methanol, like I described to you previously, to make it insoluble in water. And then we put it in a little flow cell, made a primitive spectrometer out of this grating, and then changed the amount of dissolved oxygen, or not, as the case may be, in the flow cell. And we looked to see what happened. As it turns out, my hemoglobin remained active in this silk grating. I changed the levels of oxygen. These two peaks here were suppressed. These two peaks are the characteristic optical absorption peaks of hemoglobin. That's what they look for when you go to hospital and they put that little cuff on your thumb to measure your oxygenation. And it happened repeatedly. We can change the oxygen levels completely arbitrarily and gain fairly repeatable presence or absence of these characteristic absorption peaks. Now, this was somewhat unexpected. Hemoglobin, when it sits on the shelf, even when it's sitting in something nice like phosphate buffer solution, will form methemoglobin, which is an irreversible, useless product of tri-oxygenation of hemoglobin. And it's a very bad medical condition because then you can't breathe in your blood. Not only did my blood sit in this silk, in this pattern silk, in this silk that had been dehydrated with methanol. Because I tell you what, if you have hemoglobin just sitting around in a bottle and you pour methanol on it, stuff will not work and you cannot get it back. But the silk protected the hemoglobin and let it remain active in a solution. This led us to the fairly powerful concept, I think, of being able to use natural biochemistry embedded in optics for detection. And that's sort of our vision with this material, is that we have these complex implanted biochemical systems that can tell you far better than anything else, more specifically, more rapidly, more robustly than any other test in this biocompatible material. And we've proven that it's biocompatible by putting it in some poor bunnies. We implanted it with transistors, placed this silk film with this transistor array inside the rabbit. No scarring, no swelling, no immune rejection. Rabbit was completely healthy until we killed it. We're obliged to, by the way. So this completely biocompatible material can contain whatever you like, optical structures, electronics, complex biochemical systems, and that can go into you and tell you instantaneously what is going on inside your body. We also made some fibres out of the fibre, which I feel is ironic, given that we're getting it from fibres anyway. But you know, these are very preliminary work. I just worked out an electrospinning method for extracting these fibres from an aqueous solution, and they work pretty well. We're yet to work out a way to actually handle them or kind of light into them, but that's sort of where the state of the art is right now. Anyway, I'm going to finish a little bit early today, unfortunately. But I want to get across to you the concept of biophotonic chips, integrated optics, combined with these biocompatible materials and these complex biochemistries that can, hopefully, one day anyway, that's what my funding proposal says, revolutionise pathology and be able to tell you precisely what's going on inside your body. Thank you.